Hello, everyone, and thanks for joining us today. Welcome to Meeting on the Mesa 2020 session, mRNA Process Development and Manufacturing on the Road to the Clinic. This panel will discuss the state of the art with an emphasis on CMC, manufacturing, and related topics. So messenger RNA synthesized by in vitro transcription has the potential to transform medicine. Since its first use in 1990 to express reporter proteins in mice, followed by the demonstration of antigen-specific immune responses, there's been growing interest in this technology as a versatile platform spanning many therapeutic and prophylactic fields. In the past decade especially, technology advancements in RNA biology, chemistry, stability, and delivery systems have accelerated the development of fully synthetic vaccines and therapies. This is leading to a new generation of drugs, including enzymes, antibodies, and antigens that will be delivered by messenger RNA. It can encode any protein and enables the body to make its own medicine. Messenger RNA drugs can fill critical gaps not met by traditional small molecule drugs, available biological treatments, and emerging gene therapies. Because messenger RNA can be synthesized using enzymes rather than cell culture, the manufacturing process is simpler and more scalable than other biological medicines. Now, this has been a key advantage for the rapid response to emerging threats and unmet medical needs. In response to COVID-19, for example, we've seen developers go from antigen discovery to clinical testing in a fraction of the time needed for conventional vaccine technologies. But despite the many advances in messenger RNA technology, limitations and challenges remain. We've recently seen that the ability to rapidly scale up vaccine manufacture is limited by supplies of plasmid DNA template and enzymes. Moreover, impossibly short development timelines have required a rethinking of the CMC development process. Another notable challenge that applies to all fields of use is the in vivo delivery of messenger RNA because its large size and negative charge impede translocation across the cell membrane. Various approaches to overcome this have been investigated, including delivery materials, formulation methods, and routes of administration. In this session, we will discuss the progress and outlook for messenger RNA-based therapies and vaccines, the state of the art in manufacturing and delivery technologies, and related topics pertinent to biotechnology and pharmaceutical industries. So with that introduction, now I would like for everyone to meet the panel. So we'll go around the table. I'd like to start with Andy, if you could briefly introduce yourself and say hi to everyone.
Yeah, thanks, Nate. So my name is Andrew Geall. I'm the Chief Scientific Officer at Precision NanoSystems. The company is focused on providing microfluidic mixing technologies to transition people from the lab all the way through GMP process development and release of clinical batches. We also have a proprietary lipid nanoparticle delivery technology we provide to our clients as well. I have over 20 years of industrial experience delivering oligonucleotides with lipid nanoparticles, polymer delivery systems. They've even developed antibody conjugate systems to deliver siRNA. Pleasure to be here and join in the discussion.
Great, thank you, Andrew. And then next to Frank.
Well, thank you very much for having me today. It's a pleasure to be on this panel. Frank DeRosa here, Chief Technology Officer for Translate Bio. We are a company focused on utilizing mRNA for therapeutic applications. Particularly our main focus is for pulmonary-based diseases, but we're also utilizing this technology, of course, for vaccine applications and vaccine development with our partners Sanofi Pasteur. I've been working on this particular platform in particular for developing mRNA therapeutics for about 11 years now, actually. And prior to that, many years in the drug delivery field, mostly focused on oncology applications. Thanks for having me today.
Thank you, Frank. And to you, Jeff.
Thanks, Nate. Jeff Ulmer. I've spent the last 30 years or so in vaccines research and development, beginning with Merck Research Labs, then Chiron Corporation, Novartis Vaccines, and most recently with GSK Vaccines, where I had been the head of preclinical R&D. Over my career, I've worked on various facets of vaccines R&D, but most of the focus has been on nucleic acid-based vaccines, both plasmid DNA and messenger RNA. About 2 months ago, I retired from GSK and have been consulting for biotech and pharma companies since. Thanks, Nate.
Thank you, Jeff. And Caitlyn. Oh, I think you're on mute, Caitlyn.
I am on mute. Hi, I'm Caitlyn Harvey.
Thanks.
I am the head of CMC strategy and manufacturing at Myeloid Therapeutics. I have been most recently with Bluebird, where was introduced to a lot of the bottlenecks that Nate talked about as we grew and expanded from very rare diseases into global disease groups. But now I'm at Myeloid Therapeutics, where we are using myeloid cells, which are a very versatile cell, to combat cancer and other various types of vaccines that are out there in the landscape currently. And so what's exciting about myeloid cells is that they have the capability to migrate to really anywhere in the body, and with the right effector functions can really target the key pathogens that we're looking to combat. So very excited to be on the panel and talking with everyone.
Thank you, Caitlyn. And I guess I should introduce myself. I'm Nate Spangler, your moderator. Currently, I'm the Director of Product Strategy and Innovation at Aldevron, where I'm focused on building out our messenger RNA and self-amplifying RNA manufacturing platforms. So I'm very excited and honored to be here today with this group. And so with that, I think we will jump right into the Q&A. So I'd like to start with Jeff to sort of set the stage here with some perspective, historical and future looking. You know, it was mentioned in my introduction, you know, messenger RNA technology has the potential to transform many areas of medicine, including infectious disease vaccines. How so? What is your experience and take on that?
Well, I think mRNA has the potential to have an impact in a number of areas. Possibly the most impactful would be to change the way vaccines are made. That is, because they do not require cell culture and biological systems, rather can be made completely synthetically. This should markedly simplify how vaccines are made, produced, characterized, released, etc. And this should streamline the entire R&D process from discovery through to late stage development. And in doing so, it should reduce our timelines, possibly reduce costs, and get vaccines to those who need them faster. So to me, that's the biggest impact that this technology could have. But because it mimics a viral infection and therefore induces broad-based immunity, very similar to what a live infection or a live organism would do, it also has the potential to address some of the unmet medical needs that we have with vaccines, but also to potentially broaden the target indication to areas where vaccine technologies typically haven't been used, such as gene therapy, where because RNA, like a nucleic acid vaccine, expresses the therapeutic entity protein in situ, it could be used to express biologically active molecules like monoclonal antibodies, immune modulators. It could be used to address targets like allergy, asthma, and oncology. And because of the speed with which you could respond, this really has enabled us to be able to have a true rapid response to an outbreak infection. And we're seeing this play out right now in real time with COVID-19, where because the processes and procedures are established and plug and play, we can go from sequence of the gene you want to express to a product ready to use in a matter of weeks. And as I said, this could be used to respond to an outbreak infection, but also really has enabled personalized oncology cancer vaccines, where you can go from biopsying a tumor to having a product, therapeutic product ready to administer in a matter of weeks. And this has not been possible with existing vaccine technologies. Nate.
I have a quick question for you. You know, one of the traditional barriers for cell culture processes, particularly for vaccines, was the cost of building those facilities. I remember, you know, when we worked together at Novartis, the Holy Springs pandemic flu facility was $1 billion joint venture between the government and Novartis. That's no longer a barrier with RNA, I believe.
True. And as you well know, the footprint to manufacture these products is much smaller and can be done with very low cost infrastructure, unlike the big facilities that you need, the stainless steel vats to produce cell culture. So this is a big potential step forward from a streamlining of cost as well.
Thanks.
Great. Yeah, thank you. That's you raised a number of interesting points there, and I think one of them I would like to address to Frank. So in talking about the challenges of rapid scale up, for a global pandemic, and the advantages of manufacturing RNA, and that it's a simpler, more scalable process. We're running into issues of raw material supplies and some other things. So I think, Frank, this is an area that you're familiar with and have some perspective and perhaps could add more to.
Certainly, yeah. Thanks for the question. So what's interesting about making mRNA and even going beyond into the delivery side of it, which we can talk about during this discussion, is it actually involves so many different scientific disciplines. So it involves understanding plasmid and plasmid development. It involves enzymes, and typically multiple enzymes. So how do you scale and resource those raw materials? It involves small molecules. So if you're thinking about methylating your cap, for example, you need a methyl agent. If you're thinking about even just the nucleotides, of course, those are small molecules. And then it involves the lipids if you're using lipid nanoparticle-based delivery, et cetera. So a lot of different disciplines are involved. What we have found over the years, because we've been able to develop very large-scale manufacturing in terms of single batch production, it involves really establishing strong relationships with your vendors. And sometimes it involves many vendors. So vendors just on the raw materials, and then vendors potentially on the manufacturing too, if you don't have internal manufacturing. And I don't really view vendors as vendors, I view them as partners. They're part of the team. And that's what I think is really beneficial to build, and something I hope all companies focus on with their vendors, because it takes all those different scientific disciplines, and you're not going to be an expert in everything when you're starting. So being able to build those relationships to then start to understand, what do you need as a company? What can the vendor help you with that? How can they help you develop appropriately? And so that takes some capital investment, and it takes time. And that really comes together with respect to mRNA when you build those relationships. And I'm speaking just from experience here, where now you've put in the time to develop production protocols for enzymes, purification protocols for enzymes, and then everything in between. Same with plasmids, same with small molecules. And that is what's going to afford you the ability to now take all of those raw materials and be able to now produce mRNA on the large scale. And a lot of work has to go in on that as well. How do you produce this mRNA in very high quality in single batch production, as is going to be required as we move forward through the clinic and beyond as a field, and into commercialization of the first mRNA drug product for whichever company is able to do that. So that takes time to develop those processes, both for production as well as purification. And I think the field has made tremendous progress over the last few years, and is just being driven even further now because of this pandemic. So now it's more in the limelight. And so there's a much greater emphasis on this, much greater attention brought to it, and much greater efforts, both from company perspectives as well as a capital infusion perspective as well. So I think it's going to push this field forward quite a lot.
Great, thank you. So, back to the initial question, just the broader perspective, the overview of, from your experience, is there anything that you would like to add, to that question of how messenger RNA can potentially revolutionize medicine? What are some of the things that we should be looking for and excited about, that you might add to what Jeff had shared with us?
Certainly. So I mean, really, what are we looking to do with this? There's a lot of different applications, as Jeff has mentioned. There can be for vaccine applications, oncology applications. And what we've been focused on a lot is rare disease applications, or just therapeutic in general, chronic-based dosing, et cetera. There's a large unmet medical need out there, and a lot of the modalities today don't necessarily address those needs. And this was brought up already, but capitalizing on the body's ability to use its endogenous machinery to make those proteins or enzymes or antigens of interest is critical for that. So again, all those post-translational modifications, which are so important, play a role here. Proper folding, proper glycosylation, proper trafficking. It's amazing to be able to take advantage of that. And so there's advantages, though, with other technologies out there. You think about gene therapy nowadays. Tremendous progress has been made on that front, and you're really seeing some great data come out. But there are some areas that still can't be addressed, whether it be a single dose. You can't redose. You have to get that dose level exactly right. The durability of protein expression from gene therapy is coming into question a little bit. The cassette size, if you will, you can't make every protein you need to based on AAV delivery from that. Or enzyme replacement therapy as another alternative modality. Poor uptake of those enzymes, poor glycosylation, all of these things play a role that mRNA can fill with respect to those gaps. So it's really quite a unique approach to some of these diseases where there's still unmet medical need.
Okay, great, thank you.
Nate, if I just build on that, we're also seeing, implementation of mRNA for gene editing, where we, using an LNP delivery system in the preclinical models, we're seeing, exquisite editing of genes and corrections of, disease proteins. And so that the backbone of RNA medicine, it's early days, but that's a huge opportunity, to meet unmet need. We're also seeing mRNA implemented at the cell therapy side. There are some issues and problems with delivering lentiviral systems to get CAR T cell receptors expressed. People are now exploring, much non-viral delivery options with mRNA-based approaches that are transient, and those are starting to see traction. And so there's, it's right now we're seeing vaccines, and that's being showcased. And I think people need to remember this is just the first generation of vaccines. They're already working. Over the next 5 to 10 years, we're going to learn scientifically how the preclinical models translated to, in vivo applications in humans. And so it's very encouraging that things are working, but I'm looking forward to the next 5 or 10 years as we get on that innovation curve and things will get better. We'll see, lower doses, less adverse effect profiles, and the dose comes down, the manufacturing footprint gets even smaller, and it really plays to the strengths of the technology. And the same applies in gene therapy, gene editing. The preliminary data and preclinical models looks great. We'll see early translation into humans, and then we'll see iterations of the technology to see improvements in adverse event profiles, and then in dropping the dose, better efficacy. So we're really at the forefront of something great. We saw this in DNA, but it didn't quite translate. With mRNA, it's cytoplasmic delivery, and it looks like even the early signs are very, very encouraging. And I think we could be optimistic of what the future brings.
No, thank you. I agree. And this is something that I would like to come back to in a minute, especially, not just delivery, but RNA versus protein or plasmid or viral vector, depending on what you want to deliver. But before we do that, going back to what we were touching on earlier with, scaling up manufacture rapidly in response to COVID-19, we talked a little bit about the issue with, unprecedented scale and the squeeze on supplies. But, the other challenge is timeline management and CMC development. And I'd like to go to Caitlyn on this one and talk a little bit about how you deal with that when you have an impossibly short timeline to go from development to your beginning your phase 1 study.
Absolutely. Thank you for the question, Nate. I think that, it's something that everybody who has worked in a burgeoning company in this field is used to, and it's you need to have all these answers yesterday so that you can be charging towards what your target product profile is and your critical quality attributes associated with that target product profile. But really beginning to shape what that is in terms of a platform approach, either from a vaccine or from a cell and gene therapy perspective, definitely helps in, I guess you would call it almost trimming the fat around what your expectations are for moving rapidly into a space where there is an unmet need, and being able to rapidly adapt and rapidly bring on any other types of assays supporting release or characterization of any of the materials or methods that you're using in developing and releasing that final drug product. It's something that building what that risk management ladder looks like in the what can I have now versus what can I implement later. Because again, with cell and gene therapy, there is no great model from a preclinical perspective that you can necessarily depend upon. You have to do the best that you can to get into the therapeutic area that you're trying to get into. But being able to then take that step back and say, okay, so phase 1 is really where we're going to be drawing that line on how we look at the response that we are getting from the patient cohorts that we've selected and stratified for. And then building that package of either potency, the critical quality attributes that are for, efficacious dosing, narrowing in on that efficacious dose, to everybody's point, right? To try to make the process available to the patient who is in need. And that's what I really do love about the field that we're in right now, is that all these companies seem so patient focused and patient centric, and that we're seeing the rise in targeting very rare genetic diseases, but also looking at how to treat a pandemic with these technologies is truly inspirational, and I think that it's going to continue, and I hope it does continue.
Great, thank you. I agree. So from that, talking about rapidly responding to urgent need, I'm trying to think which way to go from here. There are many interesting possibilities. So maybe just to finish on this, from your point of view, is there anything about RNA that you see as easier or more difficult when you are rapidly working towards an outcome?
Yeah, no, I think that it's one of the greatest considerations I think you can take, right? So you look at some of the standard cell and gene therapies that have really revolutionized the field, and you have starting materials to drug substances, 1, 2, 3 to drug product. And you look at that in terms of what you have to control for, from everything that Frank was saying, from custom enzymes and the release and scale of those to treat everything from a severe genetic disorder to a global population. You then look at it to what specifically Myeloid is doing in terms of using mRNA. And we have mRNA as our drug substance 1, and then we have the patient's enriched myeloid cells as drug substance 2, and we combine the two to make our drug product. So significantly shortcutting that extensive supply chain dynamic that exists in a lot of the standard cell and gene therapies that are currently out there, has, in my mind, right, and hopefully as we are entering the clinic, will revolutionize that needle to needle time to really be able to get into patient populations that, currently have no other real options out there, and get it to them quickly.
Yeah, thank you. So I'd like to go from that over to you, Andrew. You have a lot of experience developing technologies, and especially in formulating for delivery. So, when we're talking about mRNA-based vaccines and other therapies, what do you see? What's your, I guess, your perspective, and what do you see as the opportunities and challenges that make mRNA perhaps a better choice for certain uses?
Yeah, I mean, I did some early work on delivery for vaccines when I worked with Jeff at Novartis. we established a program in 2008 to look at self-amplifying RNA. And, we knew delivery was going to be one of the biggest barriers, but it wasn't going to be as difficult as DNA because you only needed to get the mRNA into the cytoplasm. So we were a lot more optimistic. Getting DNA across the nuclear membrane and into the nucleus was a significant barrier. And, Inovio has its solution, which is electroporation. And it's not ideal. It's a big device, and there are some issues with, patients being uncomfortable using it. So when we surveyed the landscape, we were looking for technologies that were highly scalable. We're Novartis, big pharma group. We're looking for things that had precedent. And we looked at lipid nanoparticles because of the work that had been conducted by Peter Collis at the University of British Columbia, developing a process called ethanol dilution. Lipids in ethanol, RNA or nucleic acid in the buffer. You mix them together, you get spontaneous encapsulation. And we thought that that might be a scalable process. And there were companies in the field working. Alnylam uses a lipid nanoparticle. The first product that was approved on Patro for them was a lipid nanoparticle formulation developed in the early 2000s. It has problems and issues, but it works, it's a product, and it meets an unmet medical need. And so there was a precedent for a process there that we felt was scalable, and I'll give a big shout out to the group at Novartis. The team was the first to publish on lipid nanoparticle mediated delivery of mRNA in mouse models, and showed surprisingly it worked very, very well. And we've seen people exploring LNPs for the delivery of mRNA vaccines. And I think of the 5 major RNA vaccine clinical trials, I think they all use lipid nanoparticles because it's felt that it's a scalable process. But scale at small scale and scaling to, global capacities, that's where the issue is going to be. How do you achieve that exquisite particle formation and encapsulation at a few liters and then scale that up to hundreds and thousands of liters? And so that's probably the biggest challenge ahead as a community. We're seeing great data in those early phase 1, phase 2 clinical trials, but the scalability is going to be pushed and accelerated, and hopefully the work pioneered by Alnylam will see us in good steads for that.
Okay.
At Precision NanoSystems, we're focused on microfluidics. That's what we believe. We have a new architecture for our microfluidic device. We've launched a GMP system, and I cannot tell you how busy we are implementing those systems around the world to help people with LNP-based COVID vaccines to get them up to scale and compliant and releasing clinical batches.
Okay, great. So a question for Jeff. Jeff, you and Andrew go back a ways. Following up on what Andrew just shared, what do you see as some driving forces behind the development of messenger RNA technology?
Well, I think, as Andy pointed out, scale is going to be necessary to meet global demand, whether it's for a vaccine or a gene therapy. And that will be driven at least in part by the dose of the RNA required. If you need hundreds of micrograms, that's a very different scale than if you need 1 microgram or less. So I think one of the opportunities we have with the technology is to identify which flavor of the RNA, and there are several that are being actively pursued, whether it's a conventional messenger RNA with modified bases or natural bases, or self-amplifying RNA. If we can reduce the amount of RNA that's required, that will go a long way to enabling us to meet the scale that's going to be required. And then, of course, there's the tolerability and safety side of things as well, because RNA is not an inert molecule. It is readily recognized by the innate immune system. We need to strike the right balance between achieving potency but maintaining safety. So there's a lot that we still don't understand mechanistically about how RNA vaccines are working.
Mm-hmm.
And the more we learn about that, the more we can take rational approaches to designing the most optimal way to achieve that balance.
Okay. And, Frank, could you maybe continue on that a bit? What do you see as the current state of the art for the mRNA-based therapies and vaccines and the challenges that remain?
Sure. So, I think everybody here is very familiar with the state of the art, certainly, and can each speak to it, but mRNA certainly has come a long way, particularly on the therapies. From my own personal experience, we set out to do this back in 2008 to use it as a therapeutic as opposed to a vaccine. And so we spent many years really optimizing both the mRNA as well as the drug product. And all those aspects include mRNA design. The analytics around characterizing your mRNA really weren't that widespread at all. A lot of these had to be developed custom. I think everybody here can speak to that, certainly. The delivery side, the lipids, the lipid combinations, those ratios, how you put those together, as Andy was saying, how you even mix them makes a huge difference in terms of the performance in vivo. So all of these things have moved forward over I'd say the last decade, certainly, and then of course the manufacturing. So these are focus areas for all of the companies out there now. Our focus has been on delivery, on the mRNA quality, and certainly on the manufacturing side of that. And what I mean by that is both scale and quality. So from a scale perspective, we've been able to demonstrate internally, at least within our company, certainly 100-gram single batch production, 250-gram single batch production, even done half a kilo now in terms of multiple proof of principle runs, knowing that now we are able to confidently say we can do that. And as Jeff was mentioning, it's all based on the amount of material you need. So because we had a pulmonary focus, and we continue to have a pulmonary focus, those doses are typically higher for a chronic-based application than for a vaccine. So the demand to put all this together at large scale, and that goes back to the original question of working with your partners out there to increase the amount of raw material needed to make these large-scale batches, all come together in terms of hopefully now pushing the state-of-the-art of mRNA forward. It's about the quality, though, as well, removing those unwanted impurities, prematurely aborted sequences, double-stranded RNA. You've heard many talk about these already. The process enzymes, all of these things, you want to have as high a quality and high purity mRNA as possible. Understanding your 5' end, understanding your 3' end. Did you methylate your cap the proper way if that's the technology you're going with? More and more now you're seeing the use of co-transcriptional capping of a cap1 type structure as opposed to an ARCA-based co-transcriptional capping. So now there have been major improvements in terms of the percentage of capping through co-transcription that really afford a much better product accordingly. From the challenges, I would say still with respect to the state of the art, because I think there are still a number of challenges, particularly with therapeutics, certainly chronic dosing, the ability to redose and maintain protein production, that translation of preclinical data from mouse to non-human primate even. There's certainly differences there. And then again, non-human primate to human, maintaining that potency, setting your therapeutic index, et cetera. All of these things are potential issues that people are working on, making a lot of progress out there. The biodegradability of your delivery system, again, primarily to Andy's point, has been primarily lipid nanoparticle based, at least in the clinic right now. All of these things are important, your DMPK profile, et cetera. And of course, safety, that is of paramount importance. I think an area of growth, Andy also alluded to this, that we'll see over the next few years, certainly, and it's just starting to come out now, is clinical efficacy, clinical data in general, safety data as well as potential efficacy. So we at Translate Bio at least released some interim data with our CF program. That's a pulmonary-based application that we were very encouraged by, but it's early days. We want to continue to generate that data. Other companies are continuing to generate that data. You've seen a lot of data now in the vaccine front with mRNA. It's very encouraging, certainly. So we're really pushing and rooting for all of our peers in the field to really succeed that, because I think it's going to really push, obviously, the technology forward a great deal and push the state of the art forward.
Yeah, and you know, RNA, it's been around for a while, right? But it's still new to manufacturers, clinicians, and so forth. So, you know, in terms of when you mention quality and safety, you know, in terms of characterization and release testing, does it seem like, you know, that, you know, what kind of progress are we making on figuring out, you know, measuring critical contaminants, and what are threshold levels for safety, characterizing the molecule itself? What are you seeing there for tests?
Is that directed to me?
Yeah, just a quick follow-up, you know.
Yeah, certainly. I mean, I think it's all moving forward. Great. I think that you're starting to get a little more consistency across the field, I would say. The FDA and more regulatory agencies are seeing more and more of these programs. So they're starting to be able to now see what's coming in and starting to set a basis associated with that. So full characterization of your drug substance, full characterization of your drug product. Again, a lot of these analytics really weren't available years ago, and most companies have to develop some of them custom, you know, proprietary based methods. But the move towards much more quantitative measures for each of these critical quality attributes is important, I think, and I think the industry is moving that way. One thing I will say that's nice about mRNA is for many of these assays, not all of them, but many of them cut across any target you're doing. So you develop, let's say, a CAP assay for one target, one program, one vaccine. You can utilize that for another vaccine target or another vaccine target, or a therapeutic target, et cetera. And that really goes along the lines of the manufacturing process itself also, this flexibility associated with that, where you develop, let's say you develop a manufacturing process for COVID, all the way through to the large scale. You can utilize that process for any other target you want. You're just changing really that plasmid DNA template sequence, as you know. So it really affords consistency also, and brings about a more robustness to the product, to the process, to the field, I think. So it'll be interesting to see how it progresses. Yeah,
indeed. So Caitlyn, on, you know, from your perspective and the things you're dealing with, you know, and talking about what it's going to take to get through your clinical stages and get your approvals, you know, what are your, you know, I guess, how can you describe the things that you've learned, and, you know, what's been helpful, you know, in terms of ensuring an acceptable CMC development, or whether it's from regulatory agencies or whatnot? What are you, what's helping you meet your impossible goals?
Yeah, it's a great question, and it's something that, as you know, when you look at this revolutionary new field, right, and you're in the you're in the cell and gene therapy space that the company that I'm currently at is in, it is remarkable how keeping up the project flow and the program flow and the visibility of that to the FDA, it really helps set the standard for what you have to keep adding on as you continue to build these clinical packages to go up for IND approval. And it is something that, you know, even 5 years ago, you know, having an interaction with the agency every 6 months seemed revolutionary, seemed like something, wow, you're getting a lot of attention, right? But now with the innovation that's happening in the space and how many, to Frank's point, custom assays are coming into play that are really helping to characterize the safety and efficacy of these drug substances and drug products, 6 months is like 3 years, right? So so the constant interaction and building that platform approach where, you know, you really are just changing, maybe not necessarily a lot around your 5' region, but you're able to utilize and change a lot within that 3' and that sequence of interest, and utilize again and again the same type of manufacturing process and supporting assays is something that is really critical in building what patients need, right? And so getting into those key phase 1 first in human trials. But, you know, as we all sit in this industry, that competitive edge still comes in, and it's that, how do you raise the bar with the agency to be preclusive, to keep yourself competitive in that landscape, which is something that, you know, we all sometimes soft shoe around, but as we are companies that are looking to continue to have a pipeline and provide really the top line therapy out there, being able to continue to develop and interact with the agency and show that you are a company that evolves and pivots rapidly based off of their suggestions, you know, does give you a niche in that space. And, you know, it definitely is, I think, what drives all of us towards that really competitive and creative innovation.
Sure. Sure, thank you. All right then, well, maybe shifting gears a little bit to Andrew, you know, talking about mRNA design, and, you know, there's many different fields of use. So, you know, everyone would have their own particular design goals. But from your experience, you know, what can you share about optimizing design for function and modulating immunogenicity, and how might this differ for vaccine versus therapeutic use?
Yeah, there's a lot of work happening in this space. Obviously, you know, the early work I did on RNA vaccines at Novartis from 2008 to 2015 was all with self-amplifying RNA. You know, there we were utilizing the ability of the RNA to self-amplify, to look like a viral infection to the immune system at the same time as launching the antigen. And so, you know, very potent in the preclinical models and very immunogenic. Also, you know, mechanistically, we published on delivery with a lipid nanoparticle, and there seemed to be two modes of action. There was the immediate response of the immune compartment sampling and taking up the LNP and the RNA, seeing it as a viral particle, getting a type 1 interferon response. And then at the site of vaccination, the RNA was delivered to the cytoplasm. It launched, began to self-amplify, express the antigen. And that combination of both led to a potent immune response. But about a week in our mouse models that we published on, that cell then went into apoptosis, a natural process of clearing viral infection, and that led to further enhancements of the potency of the vaccine. And so very complicated mechanism of action. And it goes back to Jeff's point. The more we understand about that process, the better we can get at teasing out how to optimize it, how to reduce adverse event profiles. We're definitely seeing those in the clinic. You're seeing both local and systemic reactogenicity to the RNA, and the combination in the lipid nanoparticles seems to be dose dependent, and the lower dose should help to alleviate that. So understanding mechanistically what's going on. But then in the RNA landscape, there's more than one type of RNA. Self-amplifying, and Jeff alluded to it, conventional RNA, base modified and unmodified. Mechanistically, probably all different. And understanding those individuals and how to deliver, we may end up in a place where we're using different optimization strategies for different types of RNA to get the end result in terms of immunogenicity. When it comes to, you know, gene therapy, it's the opposite, and there's plenty of data showing from a delivery perspective, those delivery systems for vaccines may not be the most optimal for gene therapy applications, where you're just looking for a protein expression of a self protein in the background of a non-immunogenic type response. And there we've seen implementations of, you know, base modification, mammalian base modifications into the RNA to try and dampen down immune responses. And so there's a lot of design happening. There's some of it's published, a lot of it's proprietary, still hidden, you know, at those companies, you know, as they develop their, you know, slant and bent on the technology. So I think it's going to be exciting to see what comes in the next 5 years as those companies start to publish data. But certainly a lot of work on mRNA design, and it really does matter for what application you're going to use it and how you design it.
Okay. Thank you. So jumping around a little bit, I wanted to go back to something that we've been discussing a little bit about, you know, scaling up for COVID-19. And this is to Frank. and a bit related to the things Caitlyn focuses on. You know, from your point of view, what do you see as effective strategies to minimize the risk with these programs that are pushing us in ways we've never been pushed before in terms of manufacturing, both in scale and timeline?
I think a lot of it comes down to parallel processing when you're moving on such fast timelines. And that's really ultimately comes down to a monetary risk, which is what you're seeing now with a lot of these organizations providing the funding associated with it. So you never want any cutting corners on quality or safety. And I think that is exactly what's happening. Everybody's following it appropriately. Nobody's cutting corners. It's just about parallel processing. So again, a huge benefit to that, to the mRNA technology is that manufacturing flexibility. You use the same process regardless of the target. So you're just changing the sequence. So having a robust manufacturing process, having robust analytics that cut across any target really allow for what I would say is an extremely fast turnaround. You've seen that now demonstrated in the field, particularly for COVID, and that can dramatically cut timelines while still not ignoring quality and safety associated with that. So that goes to both the messenger RNA as well as your delivery system, particularly if you're using a lipid nanoparticle.
Okay, thank you. So Jeff, you'd mentioned the flavors of RNA, and in particular, we have messenger RNA and self-amplifying RNA. And you know, while we're on the topic of vaccines, you know, self-amplifying RNA especially seems like a promising technology. Could you talk a little bit more about these two flavors and how you see them playing out? And you know, ultimately, you know, is there a winner or is there room for both?
Well, I think, you know, as what Andy was saying before, different types of RNA for different indications is likely. Each of these different RNAs can be tuned. The delivery systems can be tuned. So the combinatorial possibilities is quite large. But from a pure infectious disease vaccine application, there's at least a theoretical reason why a self-amplifying RNA might be desirable. As Andy mentioned, it is viewed by the human immune system and the cells in which the RNA is delivered like a virus. These self-amplifying RNAs are basically engineered RNA viral genomes in which we've removed a few of the structural genes. In place, we put the gene of interest. So when that RNA is delivered and starts to amplify, it looks like a virus. It gives short-term expression of the antigen in the matter of a few weeks, and therefore it looks like a virus and is probably treated like a virus. And in doing so, you have what is a mimic of a viral infection, very similar to what, you know, the live attenuated virus-based vaccines achieve. And these are pathogens that have been engineered in some way to make them safe, but for all intents and purposes, it launches an infection and the immune system responds very robustly. And what we're attempting to achieve with the self-amplifying RNA is to mimic that viral infection, but in a much safer way than you would with a live organism. So if we can achieve that balance of, again, potency and safety, that may be the best way to approach a vaccine application of RNA. Having said that, though, of course, for certain viral targets where antibody responses are the protective means of immunity, all RNAs produce the protein and produce an immune response against that protein. So the T cell component that you're eliciting with this mimic of a live infection may not be necessary for all infectious disease targets. So I think the jury is still out on what is most important, but the one major thing that the self-amplifying RNA might achieve is lower doses. And therefore, if we can achieve a similar level of immunity with a few orders of magnitude less RNA, that will be a very practical attribute.
Okay, thank you. So Andrew, on the topic of delivery, this is something we could probably fill a whole session discussing.
Absolutely, yes.
It's certainly a very hot topic, and I know as time is starting to run down, I wanted to make sure that on this topic, you know, from your point of view? Have we missed anything? Is there something you would like to add on delivery?
Yeah, no, we've absolutely missed something. So part of the delivery conundrum is actually stability as well. You know, you've got RNA is not DNA. It was built as a transient message. It will hydrolyze, it'll oxidize, it'll deaminate, and you know, you have to go in knowing that. And when you build the process, you know, at scale, you're going to have hold times. You're going to, you know, it's not going to take 2 hours. It's going to take a period of time where your RNA is formulated, it's encapsulated, and it's going to be at ambient temperature or 4 degrees, and that engineering space needs to be scoped out. And so you need to understand the stability profile of your RNA. And even, you know, through manufacture, and then when you come to release that product, it needs to be stored appropriately. And we're seeing, you know, all the early clinical trials now, LNP RNA formulations at -80. I saw some surprised announcement in the press that they just discovered this. It's an early technology. There's no surprise that we're at -80 because that's where things are stable. And we're going to see innovation happening. We're going to see, you know, people learn to, you know, develop formulations that are lyophilizable, that have extended stability at room temperature and 4 degrees. And you know, it'll be interesting to see what happens. You know, and then near term, we've probably got the first chance to look at all the RNA technologies delivered by an LNP, probably different LNPs, nobody's disclosing too much. But we're going to see data in the same disease target, in relatively the same, you know, phase 1, phase 2 conditions. And we'll see how the technologies stack up against each other, the 3 different types of RNA. And then we'll see how the people improve over the next 3 to 5 years, because this is just the first iteration of the technology. And the clinical learning curve has really started. And so for me, that's what's exciting. But definitely on the LNP, on the delivery side, we've got to keep our eye on RNA stability, understanding how RNA degrades and how we can influence that and slow the process down and end up at a more pharmaceutically, you know, acceptable storage conditions for long-term use. And then, of course, you know, everyone's focused on LNPs. It's not the only way to deliver things. You know, there are lots of other technologies out there. And as people get access to mRNA in a more cost-effective way, we're going to see people revisiting delivery. We saw an explosion for siRNA. Before that, DNA, mRNA, it's a different cargo. There will be different solutions. And so I think we're going to see innovation happening in the delivery space. I've done polymer work for a long time, and I'm going to be excited to see if finally we start to see those polymer delivery technologies delivering on some promise.
Yes, true. And Frank, you had something?
Yeah, sorry, I didn't mean to interrupt that. I just wanted to add to it, and I agree with that. I think you will see some polymer movement in the future, certainly. To Andy's point about stability, what a great point to bring up. And so many people just traditionally think about stability with mRNA and think about nuclease degradation, because that's obviously where it's not stable. But to your point, as a drug product, it's not typically the nuclease degradation you're worrying about. It's that hydrolysis and other pathways as well. And so it's a very different way of thinking, at least publicly speaking, that is now starting to be discussed a little bit more, certainly. And to your point again, lyophilization, I think is certainly a direction I think many companies will go in, just because, of course, the removal of your water there in terms of your source of degradation potentially.
You know, and for people that are listening, you know, on the broadcast, you know, Jeff and I, you know, published a lot in this field, 2012 to, you know, 2020, and buried in some of the book chapters that we co-authored are discussions about RNA stability. How does RNA degrade? What are the mechanisms that it would use? And starting to think about how would you go about, you know, developing assays to tease that apart. So, and there's plenty of literature out there as well.
All right. Well, thank you. Yeah, that's it's super interesting, and yeah, we could spend another hour, but for sure. Thank you. On just checking time here, I think let's go
So
to
Nate,
Caitlyn.
I have a question.
Yes, go ahead.
You're at Aldevron.
Yes.
You're right in the thick of it. How busy is it? For you, because we need plasmid and we also need RNA. So how are you coping at Aldevron? Just to give us a flavor of what it's like with the COVID pandemic.
Well, it's very exciting times for us, and the timing is fortunate in that the company is already in a very rapid growth mode. Well funded with investors. So for us, it's an opportunity to really help out in an impactful and meaningful way. Not just growing a business, but being able to do all of this and meet these challenges. And it's not just the unmet medical needs, it's now we have unmet manufacturing needs. So it's exciting to solve this problem on many fronts, for sure. Fast-paced, pressure, definitely, but it's all good. So yeah, thanks for bringing that back to my perspective and the manufacturing side of things. We're very glad to be a part of this. So. But what we do at Aldevron, we usually go to the stage of drug substance. So we get to hand it over at that point to someone, like yourself, who can say, okay, now we have to, here's where a bit of magic happens, and now it's drug product. So this is definitely, I think, a hot topic that people are going to follow closely in the years ahead, and that we're going to learn a lot about, meeting the current need for COVID. So I'd like to, again, looking at time, I think I'd like to end with Caitlyn on, hopefully our audience has gotten a sense of, all the excitement and potential here, all the things that can be done with messenger RNA in a way that hasn't been possible with other modalities. But, with all that said, it's not going to mean anything if we can't get it to the patient. So, Caitlyn, this is where you come in. Can you talk a little bit about, a CMC project manager? It's a key role in ensuring rapid progress and in avoiding delays that are going to impact, how soon a patient can get this groundbreaking treatment. What advice can you offer others managing accelerated clinical programs?
Yes. So it's definitely a dynamic space, right? And in a small company like I'm at right now, partnering is one way to do it. And so, we went and we asked the experts, and they have held our hands and helped us meet those timelines, manufacturing the drug substance. Thank you, Aldevron, for doing that. And so, it's really, and working together, right? So we then work with Aldevron with the feedback that we get from the agency to rapidly institute a new assay if it's needed, or a new consideration, or justify a specification that's on there. So hopefully we're both building our expertise to be able to rapidly continue to move these types of projects forward. And it's one of those things, I'm sure, Aldevron experienced it with having to make, up to 4 to 6 plasmids to create a single lentiviral vector to then, create that drug product. Everything will eventually become a manufacturing bottleneck. And so to be able to, have that nice iterative cycle, to be able to continue to push things as rapidly as we can to the patients that are in need is going to remain something that is integral in any of the relationships that we have from a CMC perspective. So with our vendors, with our partners, with the clinical sites, with the drug product manufacturers, it really does just build this amazing matrix that needs to have the ability to iterate across that spectrum.
Great.
That's definitely how we do it here.
Great, thank you. All right, well, I think before I wrap up, I'd like to go around the table once more, just to check in with anyone to see if, any last things to add, starting with Jeff.
Well, I've been in vaccines R&D for 30 years, touched on many different technologies, but I have to say this one excites me more than anything I've worked on so far. Cautiously optimistic.
Agreed. I might be biased, but my background is in recombinant protein therapeutics, and I have to say, yeah, I share your optimism and excitement. How about you, Frank?
Well, I just want to say thank you to you as well as all the other panelists, first and foremost, for spending the last hour chatting about this. Certainly an exciting field. I also want to say to you, Nate, congratulations. It's been amazing watching Aldevron grow over the years and how much the company has expanded and the trajectory of where they're going. And Michael, Michelle, Tom, everybody over there has just done a phenomenal job. It's very impressive. So congrats to that, and best of luck as you continue to expand.
Thank you. Thank you very much. And then how about you, Andrew? Anything else to add?
No, I'll just build on, some of the things that Jeff said. I was working at Novartis in the pharma division in 2008, and I had an opportunity to move from pharma into vaccines, leave a group of about 16 scientists. They gave me a couple of postdocs and an opportunity to start to pioneer and look at RNA vaccines. And I was a big fan right from the beginning, and it's fantastic to see how the whole field has grown, since that time and expanded, and now it's at the forefront of the battle against the current pandemic. So it's just an amazing journey over these years. I dipped out for a little bit to spend 5 years at Avidity building an antibody siRNA platform, which is now on its way into rare diseases in muscles in the clinic. And now I'm back in working on mRNA. So it's exciting to be back.
Well, glad to have you back in it. So exciting times indeed. And Caitlyn, any closing remarks?
Yeah, I mean, it's been wonderful to be on the panel, especially with all of these innovators. I hope that we get to keep championing all of our causes and getting these innovative therapies to the patients that need them, and improving quality of life and meeting the unmet medical need out there. So let's keep pioneering.
Agreed. Agreed. Well, with that, I think I will wrap this session up, though I could probably keep going for quite a while with everyone here. It has been a great discussion, and thank you very much. So, I guess just to close the session, when we're looking at, the first mRNA-based therapies and vaccines in the clinic, they're proving their safety and effectiveness. The future looks really bright, and it's going to expand to include a wider and more comprehensive range of indications. Leading the way, I think, it's likely going to be both therapeutic and, vaccines, prophylactic vaccines, mRNA-based therapies, to deliver antibodies, enzymes, and hormones. So in the years ahead, watching the progress of all these and how it's going to set the stage for so much more is really going to be exciting. So I'd like to thank the audience for your time and attention, and my guest panel for your interesting knowledge and insights. And it's been fun. Maybe we can do this again.
Thank you, Nate.
Absolutely.
Thanks, everyone.
Nate.
Thanks, Nate.